Okay, so our next speaker is Dr. Fabian Landman. Fabian works as a scientist, researcher, and bioinformatician in the Center for Infectious Disease Control in the National Institute for Public Health and the Environment in the Netherlands since 2013. In 2017, he introduced ONCT long-read sequencing in the department, and his main research interests are antimicrobial resistance, long-read sequencing, data processing, and generation of bioinformatic pipelines. The floor is yours.
Thank you for the introduction. I work at the Dutch National Institute for Public Health and Environment, and today I would like to present about our work, how we do genomic surveillance using long-read sequencing data with Nanopore for the genomic surveillance of multidrug-resistant organisms. So the goal of this study is we are initially interested in the spread of these resistance genes amongst these multi-drug-resistant organisms, among persons and in healthcare systems. So if you wish to analyze trends in these antimicrobial resistance and all these genes, you have to accurately reconstruct these chromosomes and plasmids because they are initially those genes are usually located on mobile elements, so they are transmitted between species as well. So our question was, because in 2017 we used a hybrid approach all the time, but our question is, can we actually now replace the previous method with just Nanopore long-read sequencing? In our outbreak studies, for which we use whole genome MLST scheme based approaches. So our data set was what we usually receive. So all the Dutch medical microbiology labs, they send us their isolates, which are suspected to be positive for copenamase producing or MRSA isolates. In a period of May till September, that was the selection of all our isolates. We received over 3,000 isolates. We did not sequence them all because the majority of this is our MRSA isolates. So we only did a subset of those. But everything that was submitted for CPE, CPPA isolates, we did sequence them with long-read sequencing and short-read. So that we ended up with, you can see the species, with a 350 isolates, the first set of species, very representative of what we usually receive. So in short, our method, we use a Maxwell Promega system to automatically extract the DNA. You can extract DNA of 48 isolates up in one time, so very low hands-on time. It takes about 1 to 2 hours for a run. This same DNA was already used for the rapid kit on a Nanopore 10.41 flow cell. And since we started our study set from May, this meant that all of this data is 5 kilohertz option enabled. And we did the base calling afterwards through Dorado duplex mode. And we assessed 2 different models. And one of the model, we also used a polishing step, but I will go into detail about that in the next slide. So as mentioned, the same DNA we also used for a short-read sequencing, for which we used Illumina platform. We used NextSeq 550. And this read quality and assembly was done by an in-house pipeline, UnoAssembly, which does Spades assembler. So the Nanopore, that was being assembled with 7 different long-read assemblers. So that resulted into almost 2,500 assemblies per model combination that we tested. So because we tested 3 of them, we had 7,500 assembly FASTA files that we wanted to compare to the Illumina output. So we threw all these isolates on a bunch, and we checked what coverage do you need to find the same amount of genes in the whole genome MLST schemes that you find with Illumina. And we find that from 30x coverage up, so we bin these isolates based on what their coverage was from the flow cells. We bin them together and we see that from 30x coverage and up, you find the same amount of alleles. It's stable around 1. However, if you look into the allelic difference per isolate compared to its short-read output, then we find a 40x coverage. That's when the median difference to its counterpart stables around 4 to 7 alleles. So that's why we decided we're going forward with 40x coverage for all of the isolates. This meant that we went forward in the study with just under 200 multi-drug resistance organisms. So next up, we assessed what model should we then use to do the surveillance. As mentioned, we had 3 different model combinations. So the general model at that time was super accurate 4.20 model, and we also did the alteration with the polishing step. And there was also a research model at the time available. It's called Virio, that we also included, but it was not possible to run the polishing step here. So what you can appreciate from this figure is that across the board, the Virio model, so that's the one in green, it performed the best for all species. So you can especially see in Klebsiella, you see a much lower median difference compared to the short read output. For most species, it already looked really great. For Pseudomonas, only species that still seemed a bit too much difference. But in conclusion, the Virio model here performed best, so we went forward with the Virio model. So next up, we looked into the 7 different assemblers, which of these assemblers actually performed the best. And we looked at the median and to the 95th percentile allelic difference again. So here we find that 4 assemblers perform pretty well. So that's Canoe, Fly, Long Cycler, which is Unicycler's long read option, and Miniasm. And those last 2, so Unicycler and Miniasm, they both use Miniasm, but they have a slightly different polishing step in between on their own. And we found that for Unicycler's option and Miniasm, especially Unicycler, we found the lowest 95th percentile for Klebsiella. So we concluded that was the best assembler in this case. We also performed the MLST for the different species, and almost no differences were observed here. So especially for Unicycler, Long Cycler again, and Miniasm, there was only for Klebsiella, there was 1 isolate that differed 1 allele. And for all the other species, we found identical MLST types. We also assessed the sensitivity and specificity for AMR gene and plasmid replicon detection. And here we found that Canoe and Fly performed the best for these values. So Miniasm and Long Cycler didn't perform that much less, but they, not as high as Fly did here. So that was a bit surprising for us, especially the specificity for the plasmid replicons. It was a lot lower for Miniasm and Long Cycler that we would have expected based on the previous slide. However, all complement genes, they scored 100% for Canoe and Fly. So based on these results, we think Fly was better for doing the sensitivity and specificity in the gene calling. So here's an example of in what kind of setting we would like to use this data. So again, we combined this data to each other, so we didn't assess the clusters with Nanopore only, but we threw them into the tree that's based on the short read approach. And what you can see in this figure, you can see the long read and the short read counterpart in red and green, and you can see them lying next to each other, so they cluster with their counterparts, only then a different sequencing approach. Additionally, long read data, we were able to find additional genes that we did not find in the reports for Illumina. These are MSCRAMs. It's a very nice acronym. I'm not going to pronounce it. So again, this very nice illustrates the additional benefit of using Nanopore long read sequencing. It was a bit too short notice to include data on how well these assemblies looked. But if we, so we had a few figures, but again, I couldn't add them, about the chromosomes, and in almost all cases, the chromosome is just one entire contig for the Nanopore data. So that looks amazing. In conclusion, it's possible for this data set that we could do an outbreak analysis using Nanopore-only data, except not for Pseudomonas. So the molecular typing, MLST, whole genome, MLST, the resistance and replicon calling, for you either use Fly or you use Long Cycler. If there are newer data sets or new models, you will want to validate it on your own data set. For us, it was challenging to reach the 40x coverage on the GridION and MinION flow cells. However, we also tried a different approach that was not isolates included in this study, but we used a P2 solo And on a PT Solo, you can combine even more isolates together, so you will have to need a high throughput, but you can multiplex 96 isolates together, and you will reach, that will even bring the price down by a factor, I believe, 1.5 compared to the other method. And our median coverage for CP isolates was, I thought it was the other way around, was 130. And for our merged isolates, the median coverage was 183. So well above, very comfortable above the 40x coverage that we assessed that we needed. A lower cost per isolate, as mentioned, it depends a little bit, it's about 2 to 5 times cheaper than it's for the previous approaches. And with that, I would like to conclude this study, and I would like to thank everyone inside this tiny plasmid, and then special thanks to Anthony Hendricks and Casper Germain for their contribution to this study. Thank you for your attention.